Without genetic modification, every single bite of food you have ever eaten, every grain of rice, every piece of bread, every potato, all of it was made possible by one enzyme, one single protein that takes carbon dioxide from the air and turns it into sugar. It's called Rubisco, and it is the most abundant protein on Earth. There are 700 million tons of it working right now in every green leaf on this planet. But here's the problem. Rubisco is slow, it makes mistakes, and for 30 years the world's best scientists have tried to fix it and failed until now. Let me explain why this matters. Rubisco's job is simple. Grab CO2 from the air and fix it into organic molecules. That's photosynthesis. That's how plants grow. That's how food is made. But Rubisco has a fatal flaw. About 25% of the time, it grabs oxygen instead of CO2. When that happens, the plant wastes energy fixing the mistake, a process called photorespiration. It's like a factory where 1 out of every 4 products comes off the assembly line broken, and you have to spend extra energy to throw it away. This single mistake costs global agriculture an estimated $148 billion every year. It reduces crop yields by 20 to 40%. So naturally, scientists have been trying to fix Rubisco for decades. They tried mutating the active site. They made it worse. They tried directed evolution, marginal improvements at best. They tried transplanting beta Rubisco from algae into crops. The protein wouldn't fold properly. They tried engineering the small subunit. It broke the enzyme. In March 2026, a team at Cornell and Edinburgh published a breakthrough in Science magazine. They discovered that hornworts, tiny ancient plants, have a special protein called RBCS-star that causes Rubisco to cluster into structures called pyrenoids. These pyrenoids concentrate CO2 around Rubisco, potentially boosting crop yields by up to 60%. 60%. That would be the largest single improvement in crop productivity in human history, but there was still a problem. How do you actually get this to work in food crops? The genetic engineering approach would take 10 to 15 years of development, regulatory approval, and public acceptance battles. We asked a different question. Instead of asking, how do we engineer a better Rubisco? We asked, can we find a place on Rubisco where a small molecule could bind and make it work better? Think about it. Every drug you've ever taken works by binding to a protein and changing its behavior. Aspirin binds to COX enzymes. Statins bind to HMG-CoA reductase. Antibiotics bind to bacterial ribosomes. But nobody, in 30 years of Rubisco research, ever asked, is Rubisco druggable? Why? Because the people who study Rubisco are plant scientists. The people who find druggable pockets are pharmaceutical scientists. These two worlds almost never talk to each other. We sit at that intersection. Ashibo Biophysics uses a physics-based method, the local potency density field, to map the entire surface of any protein and identify where small molecules could bind. It doesn't care whether the protein is human, bacterial, or plant. It maps the physics. So we ran Rubisco through the Ashibo engine. The hornwort Rubisco, Uniprot Q31795, the same one from the science paper, and what came out changed everything. Druggability score, 80 out of 100. High, to put that in perspective, the average approved drug target scores around 70. Rubisco, the enzyme that everyone said was undruggable, scored 80. But the score wasn't even the most important finding. It was what we found inside. 14 druggable pockets on the single chain. When we analyzed the full experimental structure, both the large and small subunits together, that number exploded to 95 pockets. 95 places where a molecule could potentially bind. But 4 of those pockets. 4 of them are discoveries that have never been reported in any scientific paper anywhere in the world. Let me show you the most important one. We call it the allosteric bridge. It sits between 3 critical regions of Rubisco. First, histidine 292. This residue is 1 amino acid away from the catalytic center where CO2 is actually fixed. Second, aspartate 324. This is in loop 6,
the flexible flap that opens and closes over the active site. Loop 6 is the structural element that determines whether Rubisco grabs CO2 or oxygen. Third, alanine 254 to arginine 258. These are in the loop 2/3 region. Another specificity determining element. For 30 years, researchers have studied these three regions independently. They mutated loop 6. They mutated loop 2/3. They mutated residues near the active site, always one at a time. Please watch part 2 of this video.